Every living organism is built from individual structural and functional units called cells. They represent the basic level at which life exists, forming the tissues and organs that make up the human body. However, there is a limit to what we can see. When two objects are placed extremely close together, the human eye eventually loses the ability to see them as separate, and they appear as a single point. This is the limit of resolution. Viewed from 25 centimeters, 2 points must be separated by 0.1 millimeters to be distinct. Because most cells are significantly smaller than this 0.1 millimeter threshold, our natural vision is insufficient. Studying the building blocks of life requires technological intervention to bridge the gap between our eyes and the microscopic world. That intervention began in 1665, when Robert Hooke examined biological specimens using a microscope he designed himself. While examining a thin slice of cork, Hooke observed a series of small, box-like compartments stacked together. He noted the resemblance of these compartments to small rooms and officially named them cells. Hooke provided the name, but modern biology requires the ability to quantify these structures. To understand the scale of life, we must calculate the specific physical dimensions of the cell. This raises a practical problem. How do you measure an object that is far too small to line up against a physical ruler? We use a method called the field of view. First, a transparent ruler is placed on the microscope stage to measure the diameter of the circular area visible through the eyepiece. If the diameter is 3 millimeters, we must convert it into micrometers for a proper scale. We do this by multiplying the millimeter measurement by 1,000. Establishing this diameter in micrometers is the foundational step. It provides the metric needed to estimate the size of any specimen placed under that specific lens. Once the field of view is known, we remove the ruler and replace it with a slide of cells, like an onion peel. Next, we count the number of cells that line up end to end along the diameter of that circular field. To find the size of a single cell, take the total diameter of the field of view in micrometers and divide it by the number of cells counted across that diameter. Let's apply this to a practice problem. If your field of view is 5 millimeters and you count 50 cells across the diameter, what is the size of one cell? Pause to solve. First, convert 5 millimeters to micrometers. 5 times 1,000 gives a diameter of 5,000 micrometers. Now, divide 5,000 by the 50 cells. The resulting estimate is 100 micrometers per cell. This formula provides a standardized method for turning a visual observation into a calculated estimate of a cell's physical size. While light microscopes allow us to estimate cell size, resolving the internal structures inside a cell requires a significant increase in magnification and clarity. For fine details, scientists use the electron microscope. These instruments use a beam of electrons instead of light to reveal structures at the nanometer scale. This image of a leaf surface was captured by a scanning electron microscope. By resolving images at the scale of a nanometer, 1/1000th of a meter, scientists can observe cellular features with remarkable clarity. As our ability to resolve these images improves, our understanding of the fundamental building blocks of life continues to expand. Let's review this exam summary. The cell is life's structural unit, and the human eye's resolution limit is 0.1 millimeters. Remember, Robert Hooke first named cells in 1665. For calculations, 1 millimeter equals 1,000 micrometers. Find estimated cell size by dividing the field of view by the cell count. Screenshot this summary for your study notes. Mastering these definitions and the estimation formula will ensure you are prepared for your exam. Every living cell operates as a busy, open factory. To stay functional, it must trade materials with the environment, continuously taking in nutrients and cycling out waste. Zooming into the factory's outer wall reveals the cell membrane. This boundary measures just 7 to 10 nanometers thick and protects the interior. Because it is selectively permeable, it acts as a bouncer, allowing oxygen to pass through while blocking others. Without this regulation, a cell would quickly be flooded with toxins or starve, making life impossible. The physical construction of this boundary follows the fluid mosaic model. This scientific framework explains how the membrane is built to be both strong and flexible. The base component is a single lipid molecule. It has two distinct ends, a blue, water-attracting head, and two yellow, water-repelling tails. These lipids naturally align into a double layer. The blue heads face the watery environments inside and outside the cell, shielding the yellow tails in the middle. This is the lipid bilayer. Large, irregularly shaped proteins are embedded throughout this lipid layer like tiles in a mosaic. These proteins act as specialized gates for moving materials across the boundary. This structure is constantly shifting. The individual lipids and proteins can move sideways, flip, and rotate within the membrane. This fluid mosaic architecture creates a flexible, adaptable layer that physically accommodates the transport of materials. These three beakers illustrate tonicity, the movement of water across the membrane based on the concentration of solutes, like salt or sugar, in the surrounding fluid. In the first beaker, we have an isotonic solution. The solute concentration is equal inside and outside. Water moves across the membrane at an equal rate, and the cell stays its normal size. The second beaker shows a hypotonic solution, where the outside solute concentration is lower. Water rushes into the cell to balance the levels. Remember, hypo sounds like hippo. The cell swells up. The third beaker contains a hypertonic solution. Because the outside solute concentration is higher, water rushes out of the cell, causing it to shrivel. Mastering these three rules allows you to predict how any cell will react when placed in a new environment. This lab setup shows a raw potato slice submerged in a highly concentrated saltwater solution. Pause the video here. Using the rules of tonicity, identify if this saltwater is hypotonic or hypertonic, and predict what will happen to the potato's weight after 1 hour. The salt water is highly concentrated, making it a hypertonic solution. Water rushed out of the potato cells, resulting in a piece that is visibly shrunken and lighter than the original. These results demonstrate how tonicity governs the physical reality of living tissues, with salt concentrations directly altering the mass and texture of the plant matter. This microscopic process drives macroscopic life. It is how plant roots pull water from the soil, and why human skin prunes in the bath. Take a screenshot of this summary for your exam prep. The fluid mosaic model on the left shows the lipid bilayer and proteins working together to maintain selective permeability. The table on the right defines the three states. Isotonic maintains a normal cell, hypotonic involves low outside solute, making the cell swell, and hypertonic involves high outside solute, making the cell shrink. Memorizing this framework provides the structural logic to answer any question about cell boundaries. Practice these definitions, and I'll see you in the next lesson. Imagine a high-tech manufacturing plant operating 24 hours a day. It requires a central command, dedicated power grids, assembly lines, and a shipping department. A single microscopic cell functions with this exact level of industrial complexity. To master your biology exams, you need to understand the logistics of this manufacturing process. We are going to map out the physical layout and the specific departments of the cell so you can visualize how it builds the components of life. Living organisms use two primary architectural designs, prokaryotic and eukaryotic. The prokaryote, like a bacterium, uses a primitive design. It lacks a defined nucleus. Instead, its genetic material floats freely in a region called the nucleoid. The eukaryote is more complex. These cells contain a true nucleus enclosed by its own membrane, alongside several distinct membrane-bound compartments that function like specialized workshops. Distinguishing a primitive nucleoid from a true nucleus is the first step in mapping cellular complexity, a detail that frequently appears on exam day. Zooming deep into the eukaryotic cell, we find the nucleus, protected by a double-layered membrane with tiny pores. Inside is the dense nucleolus, which manufactures ribosomal subunits. Once built, these units exit through the pores to work in the cytoplasm. These subunits head for the endoplasmic reticulum, a massive folded membrane network continuous with the nucleus. The rough ER is studded with these ribosomes to synthesize proteins, while the smooth ER lacks ribosomes, synthesizing and storing fats and hormones. By utilizing this two-part ER system, the cell can mass-produce the proteins and lipids that serve as fundamental building blocks for the organism. Once raw proteins and lipids are built, they travel from the ER in transport vesicles to the Golgi apparatus, a series of flattened, sac-like structures. The Golgi functions as the cell's shipping center, modifying, sorting, and packaging molecules for delivery. All this manufacturing requires a constant power supply from the mitochondria. Inside, the inner membrane is folded into projections called cristae, increasing the surface area to maximize the conversion of glucose into ATP, the cell's energy currency. The factory also needs efficient storage. Animal cells use small, scattered vacuoles for temporary storage, but a mature plant cell contains one massive central vacuole. This organelle stores water and waste, creating the internal pressure that keeps the plant firm. Without this specialized chain, from the power generation of mitochondria to the packaging of the Golgi, the cell could not sustain its metabolic weight. Let's verify your understanding with an active recall challenge. Mentally trace the path of a protein from its instructions to its final packaging. The sequence begins in the nucleus, moves to the rough endoplasmic reticulum for assembly, and finishes in the Golgi apparatus for shipping. Let's align the remaining departments. The SER synthesizes lipids, the mitochondria produce ATP, and the Golgi handles packaging. Capture a screenshot of this summary. It organizes the essential test terms, the nucleoid-nucleus distinction, the RER and SER functions, ATP production on the cristae, and the large plant vacuole. Visualizing the cell as a coordinated factory system connects these individual parts into a single logical process, the key to mastering these concepts for your biology exam. When you get a cut on your skin, the gap doesn't close because your existing cells stretch to cover it. Instead, the cells at the edge of the wound physically divide to make more of themselves, bridging the gap to seal the injury. This highlights a universal rule. All living things are made of cells, and the cell operates as the basic functional unit of life. We can see how this theory came together on this timeline. In 1838, Matthias Schleiden observed that all plants are made of cells. A year later, Theodor Schwann found the same thing in animals, establishing the first rule of cell theory. The timeline concludes in 1855, when Rudolf Virchow identified the mechanism for how life continues. He proved that new cells arise only from pre-existing cells. Every single organism, from a single bacterium to a giant tree, relies on this continuous chain of multiplication. Understanding this rule allows us to explore exactly how these copies are made. This microscope image of a plant root tip shows a dense matrix of cells at the growing end, constantly forming new ones. These cells are undergoing mitosis, biology's primary copy machine, built for general growth, tissue repair, and body maintenance. Let's break down the mechanics. We start with a single parent cell. Inside its nucleus, we can count exactly 4 distinct chromosomes. In mitosis, the parent cell goes through a single division process. It stretches and splits to form 2 completely new daughter cells. Because this is an exact copying mechanism, each new cell retains exactly 4 identical chromosomes. By maintaining the exact chromosome count every time a cell splits, mitosis ensures your genetic information stays perfectly intact across your body, whether you are growing taller or healing a scrape. There is a second type of cell division called meiosis. This process only happens in reproductive organs, specifically to produce gametes like sperm, eggs, or pollen. The mechanics here are different. Instead of a single split, the parent cell divides once and then immediately goes through a second consecutive division. Because of that double division, we end up with 4 distinct daughter cells. And if we look inside, we see they hold half the original genetic material, just 2 chromosomes each. If gametes didn't have their DNA, every time an egg and sperm fused, the offspring would double their chromosome count with each new generation. By cutting the count to 2 initially, the total is restored back to the original 4 during fertilization. Meiosis deliberately sacrifices identical replication. By mixing and halving the DNA, it generates the genetic variation required for sexual reproduction to work. Both processes run on strict tracks. When mitosis loses its brakes, cells divide uncontrollably, resulting in a tumor. A meiosis error operates differently. If chromosomes fail to divide evenly during the second split, gametes get an abnormal count. This imbalance leads to genetic disorders. Let's test your understanding. Take a look at the prompt on the screen and answer. Does a growing root tip use mitosis or meiosis, and how many cells does it produce? As this microscopic image of a dividing root cell shows, the chromosomes are splitting symmetrically into two identical halves. The root tip uses mitosis to grow, producing exactly two identical daughter cells. Let's consolidate this into a study guide. Mitosis is for growth and repair. It requires one division, yields two identical cells, and maintains the chromosome count. Meiosis is for sexual reproduction. It requires two divisions, yields four distinct cells, and halves the chromosome count. An easy way to remember this is mitosis makes twins, meiosis makes eggs and sperm. And every single cell produced on this chart is living proof of Virchow's rule. All life stems directly from pre-existing life.
Hope this video has provided the deep understanding about the chapter. Here is the high-level summary of the overall content summarized for you. Any individual cell is like a microfactory of its own. The cell is the structural and functional unit of every living being. For example, our human body is made up of roughly 30 trillion cells, which representing around 200 different cell types. Let's have a brief overview and get into the details in a minute. 1. All living things are cell-based. It means, from single-celled bacteria to multicellular humans, the cell is the basic unit of structure. 2. The nucleus acts as the control center. It houses DNA and provides the coded instructions for all cellular activities and inheritance. 3. Organelles functions like specialized factory departments. Here, mitochondria generate ATP, while the endoplasmic reticulum and Golgi apparatus manufacture and package products. Hope you knew that ATP is the acronym of adenosine triphosphate, which is the primary energy currency of all living cells. 4. Membranes serve as selective gatekeepers. The plasma membrane regulates the movement of substance via osmosis and diffusion to maintain internal balance. 5. Life continues through controlled cell division. Processes like mitosis allow organisms to grow, repair damaged tissues, and replace old cells. Thanks you.